Thank you very much. It's nice to have been able to meet and talk to a lot of people in the audience, all of whom are far smarter than I could ever hope to be. And I think it's so exciting how the field is moving so quickly, as you heard from the previous couple of talks. So we've heard about these pillars of aging, these fundamental aging processes, and you've seen various diagrams of them. I'm showing you a slightly different diagram. I think very simply, I like to think of 4 pillars of aging. People have up to 13 pillars of aging, but the key thing is that they're all highly interlinked, what we're beginning to call now the unitary theory of fundamental aging processes. So we've heard about inflammation. This occurs at sites of changes that occur with increasing age, but also occur in younger people in the context of chronic diseases that are related to an acceleration of action of some of these fundamental aging processes. And the kind of inflammation is chronic, low-grade, and sterile under most circumstances, that is in the absence of bacteria and fungi, and tends to be associated with fibrosis. The second major category of fundamental aging processes are changes in large molecules and changes in intracellular organelles. So there are profound things that happen with DNA. There are changes in epigenetics, this epigenetic clock business. There are changes in telomeres, the ends of DNA and how they function. There are changes in satellite regions. There are DNA strand breaks. There are all kinds of things that can occur with DNA, not only with aging, but in the context and sites of etiology of multiple diseases, even in children. There are changes that occur in proteins. We've heard about mTOR, which is a master regulator of protein production. And some of these things that occur include what Anna Maria Cuervo and others have shown with respect to failure to remove proteins, production of proteins that aggregate together. This is a basis for Alzheimer's disease and Parkinson's, for example. Misfolded proteins. There are problems with sugars. So there are advanced glycation end products that form because of reducing sugars. Mehmed Khan has worked a lot in this area, where reducing sugars react with proteins and DNA backbones, forming these advanced glycation end products. There are problems with lipotoxicity and fatty acids that are saturated, production of ceramides, prostanoids, and so forth. There are problems with intracellular organelles, mitochondria, lysosomes, and others. There's stem and progenitor cell dysfunction that occurs with increasing age, and there's cellular senescence, which Richard introduced. And it appears that these things are upstream. The geroscience hypothesis that was first put forth by Gordon Lithgow, who is now at the Buck Institute, was at Manchester before, and then made popular by Felipe Sierra and Brian Kennedy and others, that these fundamental aging mechanisms may be root cause contributors to the bulk of the diseases and disorders that account for most morbidity, mortality, and health expenditures. Again, from children through to adults, these processes begin operating at or before conception. But they contribute to development of the geriatric syndromes, things like frailty that we heard about, age-related muscle impairment or sarcopenia, immobility, malcognitive impairment. We've heard that aging is the biggest risk factor by far for most of the major diseases that account for major issues. Your risk of having a heart attack or stroke is increased 2 to 4-fold if you have a high blood pressure, high blood sugar, high cholesterol, positive family history. But if you're 85 as opposed to 30, your risk is increased 1,000-fold. If you put these things on a bar graph, a histogram, and you have just chronological age, as imperfect as that is, as a risk factor, if you accept it as a risk factor, for about the 200 most common chronic diseases, you don't even see the other risk factors because the bar for aging is so big. Then there's what we heard from Janet and others about this decreased resilience, decreased ability to respond to infections, to wounding, to chemotherapy or other kinds of drugs. And it appears increasingly that these fundamental aging processes drive those. So it's important that these fundamental aging processes are highly interlinked. It's appearing increasingly that if you target any one of them, you affect all the rest in a positive way. So we've got around a dozen or more broad categories of interventions now. and some of these, within these categories, there can be hundreds of drugs, like literally there are hundreds of senolytics now, for example. But it appears that if you use any one of these approaches, you tend to affect the rest. Rapamycin, for example, that we heard about, it inhibits what I'll mention in a moment is the secretory state of senescent cells. Metformin does the same thing. It turns out that if these sirtuin agonists or NAD are highly linked with senescence. We heard about NAD. Low NAD levels result in increased reactive oxygen species that drive senescence. Senescent cells, in turn, produce a factor that increases an enzyme on the outside of macrophages that degrades NAD. So there are these cycles, there are these circles amongst these fundamental aging processes, and intervening in any one of them tends to affect the rest. So there may be ways we can combine interventions. We don't know if they'll be less than additive, or more than additive. But what will be extremely promising, I think, in the future will be taking disease-specific interventions and coupling them with interventions that target fundamental aging processes, because there's increasing evidence, most of it unpublished, unfortunately, at this point, but will be published soon, showing that if you have a drug that, say, affects pulmonary fibrosis, for example, a little bit, or something called HFpEF, where the heart gets fibrosed. If you use a drug like current drugs that are used to try to reduce the fibrosis in those conditions a little bit, if you combine them with senolytics, drugs that kill senescent cells, and the senolytics have a little bit of an effect, when you combine them, the effect is much more than additive. It's synergistic. So I think in the next 3 to 5 years, not only will we have these interventions that target fundamental aging processes, we're going to move into combining disease-specific interventions with interventions that target fundamental aging processes in clever ways to try to treat heretofore untreatable disorders. So senescence we've heard a bit about. It tends to be a response to damage in general. It was first discovered by Hayflick and Moorhead, first published back in 1961, when they found that repeatedly replicating cells, some of them don't die, but they do stop dividing. They slow down and then they stop dividing, but they don't die. In fact, they're resistant to dying. And they become larger a lot of times than normal cells. They're metabolically very active, and that was the discovery of senescent cells. It's since been found that all kinds of damage signals will make cells become senescent. Not only repeated replication and things that drive repeated replication, but cancers and cancerous mutations and genes that go up in cancer will do it. Damage-associated molecular pattern factors, things like contents from disrupted cells, like DNA that gets into the blood, will induce senescence. Inflammatory mediators, we've heard discussion about TNF-alpha and so forth, will induce a cell to become senescent. Metabolic dysfunction, high glucose, having 33 millimolar glucose is sufficient to drive cells into senescence. Low oxygen, high oxygen can induce senescence. Mechanical and shear stress can do it. This is the basis for senescent cells forming in the knee joint in osteoarthritis, or on the venous side of arteriovenous fistulae that are used for renal dialysis patients, or at bifurcations of blood vessels. Pathogens will do this. I'll talk a little bit about coronavirus. That can cause senescence and make senescent cells that exist worse. And senescent cells produce factors that make normal cells susceptible to coronavirus. And this is true for bacteria, fungi, all kinds of other viruses. And then various drugs can drive cells to become senescent, like chemotherapy drugs or radiation and so forth. So there's no good single marker of senescent cells. There's debate in the field at times about whether a cell is in fact senescent. So we have to combine markers at the moment in order to really say whether a cell is truly senescent or not. Senescence is a cell fate, like replication, differentiation, or apoptosis. It's the fifth cell fate. A cell integrates these signals coming in, indicating there may be damage, and makes a fate decision to become senescent or not. It takes, at least in culture, a week to 6 weeks for a cell to become fully senescent. So it's much slower than the other cell fates to develop. This means that getting rid of these cells can be done intermittently. So if there's a continuous drive to produce senescent cells, unlike other geroscience interventions, you can give these agents in a hit and run approach because they don't divide. Once they're gone, and they take a week to 6 weeks to reform. So in the clinical trials underway of these agents now, if there's a continued impetus for new senescent cells to form, like high-fat feeding or diabetes or something, The drugs are given intermittently, at least in these trials. And in experimental animals, that appears to work better than giving these drugs continuously. So this is much more like using an antibiotic in a hit and run approach. And we're no longer talking about a one drug, one target, one disease thing. As the previous speakers have indicated, now what we're doing is targeting these fundamental processes. In the case of senolytics and so forth, we're going after a cell type, much like bacteria. And then we're going after many, many conditions, like bacteria can cause a urinary tract infection or pneumonia or an abscess or whatever. I'll show you in a moment that senescent cells and some of these other fundamental aging processes underlie mechanistically a lot of disorders. Some senescent cells, not all, 30 to 70% can acquire what's called a senescence-associated secretory phenotype. But it's not all of them. It's 30 to 70%. And these are the bad guy senescent cells. They're producing proteins and peptides that are inflammatory, that attract, anchor, and activate immune cells, that spread senescence, both locally and in an endocrine manner to distant, previously non-senescent cells. And in addition to proteins and peptides, these cells produce a variety of reactive molecules. Reactive metabolites are producing prostanoids and bradykines and ceramides and all kinds of things that cause dysfunction. They're producing what we call non-coding nucleotides, so things like microRNAs, very importantly, mitochondrial DNA. They release that from the cells, which causes widespread dysfunction and is responsible to a large extent for rejection of organs that are transplanted. And I'll come back to that. Senescent cells are resistant to dying because of these senescent cell anti-apoptotic pathways, or SCAPs. So there are at least 9 of them. So senescent cells can accumulate in human tissue with aging, even in relatively healthy individuals, but the sicker a person is, the more there tends to be a correlation with an increase in senescent cell burden. So very healthy individuals may not accumulate senescent cells till fairly late in life. These are relatively healthy individuals because they're donating a kidney. In some cases, grandmothers donating their kidney to their grandchild, so they're guaranteed healthy. And what we do is look at their tissues, and we find that there's an increase in senescent cell burden across multiple tissues with increasing age that can occur anytime between the 60s and 80s, depending on the individual. And in some individuals, it occurs quite late, as I mentioned. But in the context of illness, senescent cells accumulate much earlier. For example, in the context of obesity and diabetes, senescent cells form in adipose tissue in children. Senescent cells can have beneficial functions as well. They can protect against cancer because a cancerous mutation can drive a cell into senescence, so they stop dividing. They form in the placenta, and their secretions in the last 5 days of pregnancy are what drives the baby through the birth canal. And it turns out that this is the spread of senescence from the placenta to the mother in some cases can cause a syndrome called preeclampsia. And that's a senescence-driven thing coming from those senescent cells in the placenta, affecting the endothelial cells in the mother. Senescent cells can be important in early activation of the immune response, in stages of wound healing and tissue repair. So you don't want to interfere with cells becoming senescent or stop them becoming senescent, because you cause cancer if you introduce mutations that stop cells becoming senescent. But once they are senescent, if they're there and they're the bad guy cells, and if they're not removed by the immune system, you need to find other ways to get rid of them. So one thing we did was to transplant very small numbers of senescent cells into the peritoneum of mice, such that 1 in 10,000 of these transplanted cells was a senescent cell. And this resulted in frailty in middle-aged mice, and earlier death. And the earlier death was caused by all causes that mice die from. So this is one end of cost postulates. If you add something, do you cause a problem? Indeed, that appears to be the case. And as I mentioned, senescence spreads. So if you label these transplanted cells and you can use a light signal and you can see where they are, they tend to stay put if they're transplanted into the peritoneum. Actually, they migrate into the fat tissue in the peritoneum. But you find that the recipient's own cells start becoming senescent in their arms and legs. So senescence spreads in not just a paracrine, a local, but also an endocrine manner. And this becomes important with transplantation. So we work with Stefan Tullius at Harvard, who transplanted hearts from young mice to young mice and old mice to young mice. And we found that the hearts transplanted from the old mice caused, that have a cargo of senescent cells, caused organs at a great distance in the young mouse to fill up with senescent cells, and furthermore release this mitochondrial DNA, which activated the dendritic cells that you heard from Janet about in the draining lymph nodes and provoked graft versus host disease. And one of the things the surgeons know is you don't transplant kidneys from older people into younger people unless they're live-related donors. In the US, if people die in a car crash and have signed their donor card, they throw away the kidney if the person who died is over 50. And we're throwing away 35,000 kidneys a year. So we're trying to figure out ways to remove senescent cells to prevent that from, to allow this transplantation. So initially we used a hypothesis-driven approach based on an important paper by Ned Sharpless, who's now director of the National Cancer Institute, who showed in 2004 that caloric restriction, which improves health span and delays death, is associated with a decrease in senescent cells. So we began to ask, is this causal? We tried for many years to develop drugs that would selectively remove senescent cells. Then it hit us. Why are senescent cells surviving despite the fact, the 30 to 70% that have this SASP, why do they survive despite the fact they're killing the cells around them? What protects them? And it made us think of what happens with B lymphomas and chronic lymphocytic leukemia, where you see the same thing. So we asked, using bioinformatics-based approach, using proteomics and transcriptomic data, are there pro-survival pathways that senescent cells use to prevent them from committing suicide, from succumbing to the things that they're using to kill the cells around them? And in fact, we discovered there isn't just one pathway. There's a whole series of them. There are at least 9 of them, these SCAPs that I talked about before, that form a network. And so we used RNA interference to test key nodes on that network, and we found that different kinds of senescent cells depend on different pathways or combinations of pathways to defend themselves against the things they're using to kill other cells. So human fat cell progenitors, which are amongst the most abundant senescent cell type that can form in older humans, depend on Src kinase-related pathways, dependence receptor pathways, for those of you who are aware of what that is. And then endothelial cells from humans, blood vessel lining cells, depend on BCL2-related families for their survival. So different kinds of senescent cells depend on different factors. We, again, went back to the computer and used programs to look for drugs that are short-acting, because we wanted to use them in a hit-and-run approach, and are approved for use in humans or natural products to begin with. We found about 40 things. They turned out to be senolytic. We decided to focus on a couple of them, dasatinib and quercetin initially. Since then, many other senolytics have been found using that hypothesis-driven approach, and now high-throughput screening approaches. If you transplant senescent cells into young mice, into younger mice, and they're labeled so you can see them versus non-senescent cells, if you treat with these agents, you kill the 30 to 70% of senescent cells that have a SASP. You do not kill senescent cells that don't have a SASP. If you give a blast of radiation to a mouse's leg so it has trouble running on a treadmill after a couple of months, all you have to do is give one treatment in that animal's life, and it's able to run on a treadmill the same way as a mouse that wasn't treated. So once these cells are gone, they're gone unless there's a continued impetus for their formation. Those transplanted animals, if we treat with senolytics, we prevent them from becoming frail, so their grip strength, their ability to run on a treadmill, the various kinds of things that Janet was talking about with related to frailty are alleviated. In the heart transplanted animals, if we treat with senolytics, the old animals that are transplanted with old hearts die early. Animals transplanted with young hearts don't. And if we treat with senolytics, where either the donor, the recipient, or the host, we're able to reduce that death. If we infect mice with a virus related to human coronavirus, old mice all die very quickly, young mice don't. If we genetically or use different, remove senescent cells, or we use different senolytics, we're able to reduce their death considerably. And this is the basis for the clinical trials now underway with senolytics for coronavirus. So there are at least 50 or 60 conditions now that senescence appears to be related to. And in mice, But who knows whether they'll work in humans, because humans are not big mice. There are some humans who are big rats that I know, but we found in an early clinical trial, very preliminary, that if we take fat biopsies and take blood at day 0, give senolytic agents that have a very short elimination half-life for 3 days, and then after 14 days, well after these drugs are gone, we rebiopsy the adipose tissue of these obese diabetic individuals who have a lot of senescent cells. We actually do clear senescent cells from humans using the same drugs as in mice. We've been developing composite scores of blood factors that relate to senescent cell burden, and those decrease in these kinds of subjects. We did a very early trial in idiopathic pulmonary fibrosis, a phase 1 trial. There are all kinds of problems with these trials. They're not controlled. There can be placebo effects and all kinds of things, but we found decreased frailty in these patients. Idiopathic pulmonary fibrosis is a fatal disease in humans that's associated with cellular senescence in the lung and severe frailty. So this is the basis for the phase 2 trial currently underway. At the moment, in Mayo and the consortium that we work with that includes a variety of other institutions across the US, we've got a total of 27 trials underway, 15 of which are with Senolytics. Other trials are things with rapalogs, with metformin, with NAD precursors and other things. But amongst the trials with Senolytics are trials for elderly women with frailty and a gait speed of less than 0.6 meters per second who have a 2-year life expectancy. It's much worse to have a gait speed of 0.6 meters per second than most cancers. So those trials are underway. There are studies 3 studies in Alzheimer's disease that are, one of them has shown some very, very early promising results, but that was only just in a few subjects as a pilot trial. There are trials underway in diabetes and kidney disease. There are trials underway in bone marrow transplant survivors. These people have had a high dose of chemotherapy or radiation. They develop an accelerated aging-like state 3 to 5 years after their bone marrow transplant if they don't die of graft versus host disease or recurrence of their cancer. And they're getting accelerated Alzheimer's disease, diabetes, frailty, heart attacks, etc. We've got a trial underway with St. Jude Children's Cancer Hospital. They found that kids who've been treated for leukemias or lymphomas before the age of 10, many of them when they're in their 30s or 40s look like they're 70. They're dying of Alzheimer's disease at age 35 and that kind of thing. They have diabetes, they get second cancers, they're getting heart attacks. And with them, we biopsied their tissue and we found the degree of frailty they have correlates with senescent cell burden. So there's a National Cancer Institute trial underway now comparing 2 different senolytic regimens with placebo in those people. I mentioned the pulmonary fibrosis. There are trials that Sandeep Khosla is running, who many people in the audience know, looking at age-related osteoporosis. That trial is 2/3 done. There's a trial at the Stedman for osteoarthritis. There are 3 trials for coronavirus, hospitalized, outpatient, and nursing home patients. There's an interesting trial. We're going to be part of the Axiom launch in February. So the astronauts are having their blood taken before and after they come back. And it's already known that astronauts have a lot of DNA damage on their return. If you look at their urine or blood, you find incredible DNA damage molecule, amounts of DNA damage reflective molecules. Astronauts, pilots, and flight attendants are dying earlier of age-related diseases in other groups. They're getting Alzheimer's 5 years earlier than other groups, for example. It may be more than space radiation. There may be zero gravity involved because of the stresses on the cell and high G forces, as well as the high CO2 in the space capsule. So NASA's very interested in this, and especially for flights beyond the Von Allen belt, where there'd be solar radiation, which we found working with Brookhaven. Really, like heavy iron radiation, it takes 1/20th the dose of that to cause a cell to become senescent as X-rays. So these are very, very dangerous. And the Mars mission will not be possible unless we're able to deal with this. So on this flight, they're also taking senescent cells up with them, non-senescent cells, and seeing if they become senescent. So they're changing the medium, and we had to develop hollow plasticware and everything, very complicated. So in conclusion, persistent senescent cells appear to cause inflammation, fibrosis, progenitor cell dysfunction, spread of senescence. I didn't have time to go into all of it. There appears to be a threshold burden above which senescence causes problems because the rate of formation of new senescent cells exceeds their rate of removal by the immune system because senescence spreads. Senescent cells also interfere with immune function, and immune cells become dysfunctional, especially NK cells, as Janet mentioned. So there's a threshold effect, and you can show that in transplantation experiments. And this may be why, this may contribute to things like coronavirus causes cells to become senescent. Coronavirus produces factors that cause non-senescent cells to have an increase in ACE2 and TMPRSS2, which import coronavirus, and decreases innate antiviral defenses within cells. So all of these things are highly interlinked, and there are thresholds that you have to exceed. So if there's an existing burden, and then you give chemotherapy on top of it, you can give a smaller dose amount of chemotherapy will cause a greater problem in an older person than a younger person because you're adding these things together. Hit and run approaches appear to be effective in animal studies where you give these drugs periodically as opposed to continuously. They look very good in mice. That doesn't mean they're going to work in humans. We really don't know. There's anything that sounds too good to be true usually is. I'd sound a real note of caution. I lie awake every night worried about our patients on the clinical trials. I find it hard to sleep. So far, and I'm touching wood, we haven't seen major issues, but it doesn't mean they won't occur. So we really have to be careful about this. And I'll conclude by thanking a lot of people far smarter than me who did a lot of this work, especially Tamara Ciccone and Izu, who we worked together to come up with Senolytics. And there are many other people, including many people in the audience, who've helped us and are many funding groups. And I'll conclude by saying what a great honor it is for me to meet Nir Barzilai in person. I've always dreamed about this. I've heard many things about him. Some of them have been good. So with that, I'll pass over to Nir.
Thank you, Professor Kevin.